The title is actually on hunger, appetite, and food choice. A surprising menu is born in the gut-brain axis. I'm really hoping that you will enjoy this symposium, and I think the topic is very timely and very lively. And the basic idea here is to try to understand what is the neurobiology behind appetite and food choices, looking at actually different techniques, but also different models. I think this is very interesting. I will start right away with the first speaker. Speakers have 15 minutes, I remind them. And the first speaker is Suzanne Dixon. She's a professor of physiology and neuroendocrinology at the University of Gothenburg in Sweden. And she will give a talk, and the title is The Aperitif, the Neurobiology of Hunger and Appetite. Thank you, Suzanne.
So, thank you, Francesca. So it gives me such joy to be here. It's so wonderful to visit this city and be part of this symposium. I'm very much looking to this gut-brain feast, for which I seem to be serving the aperitif. So I invite you for some port. And actually, because I'm more of an appetite researcher, I also invite you for a little Portuguese aperitif. Enjoy. So yes, I'm going to be talking about the neurobiology of appetite and hunger. I have nothing to disclose. So you're probably wondering, what's the difference between appetite and hunger? Are these not just a sort of rebranding of the same thing? Well, the thinking is that hunger, in order to feel hunger, you really have to be in energy deficit. And it's very important then to eat calories. And all foods really taste good when you're hungry, even a simple cracker bread. Appetite, however, is more associated with anticipated pleasure, where you're really expecting to eat something that you find especially palatable and is more directed at specific foods. So dogma would tell you that really the hypothalamus and brainstem are really involved in energy balance regulation, and they're very much more linked to hunger and energy deficit, whereas appetite is much more a function of the reward system, that we're motivated to find palatable foods, of course, when we're experiencing appetite. But I've drawn an extra arrow here just to indicate something that's rather important, and that is that, of course, when we're hungry, the reward system is engaged. We want to search out, we're very motivated for foods, all foods, and including rewarding foods. So hunger also then engages the reward system. So the point is that for hunger and for appetite, obviously there's some overlap. The systems that are engaged when we're hungry are of course similar, but also that distinct pathways may also be involved. So whether we're eating for hunger or for appetite, there's a lot of factors in our environment, intrinsic signals that are important. One of these, of course, is just food availability. So, you know, if you're expecting a very nice meal, hunger can actually be something quite pleasurable. We've all been there. We've turned up at the lunch seminar with the promise of sandwiches, and all the students got there first and ate them all up, and we have to sit a whole hour feeling hungry, knowing we're not going to get any food, and that is, of course, rather aversive. Cues in the environment that predict food ability are also important. For humans, it's the sight and the smell of foods. Animals rely more on smell, of course. I'll be coming back to this point in a moment. Then we have intrinsic signals, we have hormones, we have metabolites. And I've spent quite a lot of my career studying this hormone, ghrelin, which is often referred to as the hunger hormone. Ghrelin is released before meal times. you have high levels. That's partly why it's called a hunger hormone. We've actually found that ghrelin can also be released at times when you're not feeling hungry, for example, when you're anticipating even palatable foods. So just to introduce you to the idea that it might not only be a hunger hormone. And in fact, we can then ask the question, could ghrelin also be an appetite hormone? And perhaps that ghrelin and external food cues could even engage similar neurobiological pathways. So again, my lab's contributed to a lot of the boxes that you see here over a long time. So ghrelin is engaged, of course, before meals for food anticipation, for food motivation, for the unpleasant feeling of hunger. And during meals, it determines our meal patterns, so how much, which foods, and when we eat. But to add on to that, it's also true that ghrelin has a role in appetite. So before meals, it's involved in the anticipation of treats, for palatable food searching, for being motivated to find treats, and is also implicated in overeating behavior and making unhealthy choices and eating when satiated. So it's also involved in eating that escapes metabolic need. So we know quite a lot about the brain pathways engaged by ghrelin. I've made a little schema here where you see some key areas that are involved in appetite. The stars represent areas where the ghrelin receptor, called GHSR, is expressed. So you can see, for example, there are receptors in the hypothalamus, especially the arcuate nucleus. This is an important area that I'll be coming back to. And some brainstem areas, including the LTG that feeds into the VTA to control this important dopamine projection to the accumbens that we showed some time ago, is also activated by ghrelin. This is a very old slide, and I just show you it because you can see where ghrelin is causing activation of cells in the arcuate nucleus. This is the arcuate nucleus here. This is even before ghrelin was discovered, when we used to work with ghrelin mimetics. And these dots here are cells that are activated that express phosphoprotein in response to this compound. These are cells activated essentially by ghrelin. And we know that, some subsequent in situ hybridization studies, we could identify, at least to some extent, what the cells are that are activated in this region. And we know that they include an orexigenic, a feeding group in this area called neuropeptide Y, and NPY is actually co-localized with another very important orexigenic peptide, which is called AGRP. So these orexigenic NPY AGRP neurons are a target for ghrelin in the arcuate nucleus. So then, what about food cues? Our question is really, what is the neural circuitry that's engaged by food cues, and could it be similar to ghrelin? They're both orexigenic stimuli. And another question was really whether food cues could even be triggering ghrelin release. So to explore this, we set up a new paradigm. So we trained animals to like the taste of peanut butter. Now, believe me, mice love peanut butter. They really love it, and they will eat masses of it. So we trained them to like it, and then we put the peanut butter in a little Eppendorf tube. We put it inside, a little tea strainer that you make your tea with, and we suspended that from the cage top of inside a cylinder. And so half the animals had tasted the peanut butter, they knew how yummy it was, and then we are essentially exposing them to the smell of the peanut butter. The other half had never tasted this peanut butter. And in fact, they don't show any of the responses that I'm going to show you for the rest of my talk. I don't know if the video here works. We see what happens. You can see at the back of the cage here, there's a little ball. Here's the cylinder. Here's the rat. And there's no sound. Sorry about that. But you can see the animal is desperately trying to access this little bowl with the smell of the peanut butter. This is, of course, a familiar animal. And it will spend 30 to 40 minutes doing this. And after this time point, it gets completely fed up. And it's a bit like when you really fancy some chocolate and you know it's there, the presence is there, but you decide not to eat it, and then you go off and eat more of your regular bread or cheese or something. So you eat calories, but not necessarily of the thing that's cued. And that's exactly what these rats do. They overconsume their regular chow when they get frustrated and not being able to access the hidden peanut butter. And these are the data here. We have repeated this in several studies. So in orange here, these are the peanut butter familiar animals presented with the cue. And at 1 hour time point here, remember, this is when they're going crazy trying to get access to the peanut butter. Even then, they're overeating their regular chow. And here you see, even at 6 hours, they're still overeating. What was also curious is that these animals, we measured their ghrelin levels. Remember, these are fed animals, so their ghrelin levels are not necessarily high. And when we exposed them to the peanut butter cue, their ghrelin levels increased. It's probably a subject for a future PhD student, but essentially we're showing that olfactory food cues are triggering ghrelin release from the stomach, which can then, of course, operate as some kind of feed-forward mechanism to amplify the erexogenic signal. So we don't know how this signal actually arrives in the stomach. Be curious to hear your thoughts on that. The motivation that I showed you is quite difficult to quantify, so we actually set up yet another paradigm. It's quite simple. It's an open field paradigm, and we put a little bait in the middle. So we secure the tea strainer to the bottom of the cage, and it's either not got any peanut butter in it or it's got peanut butter in it. So it has a cue or no cue. And the two graphs on the left are information which are consistent with increased motivation. So those animals that are familiar with the peanut butter, they have increased contact duration with the ball in the middle, and the percentage time in contact with the ball is also increased. There was no change in anxiety-like behavior in this environment. So this tells us that we've been able to show in a different paradigm that just having the presence of a familiar peanut butter cue in this environment makes the animals go and eat. So what about the circuitry involved here? Well, here's our own old friend, the arcuate nucleus that I showed you earlier, and we see FOS activation here again. And again, this is the animals that have been exposed to the peanut butter cue that are familiar with its taste. And we actually screened the whole brain, and I have to say this was the only area that clearly popped up in our studies where we saw a big activation in response to the peanut butter cue, and we've quantified it in this graph here. So the arcuate nucleus in the hypothalamus, curiously, pops up as a very important area for food cues. We went on to try to figure out what these cells are, and we've used RNA scope, where you can do maybe 3 different in situ hybridizations, essentially, on a single section. So in this particular section, so we've looked at a lot of different sections, and we have approximately per section about 80 HRP neurons, about 120 ghrelin responsive neurons, and about 100, say, FOS positive neurons. And then when we looked at co-localization, we were a wee bit surprised to discover that only 20% approximately of the cells activated by the food cue that express this FOS could be identified as these erexogenic AGRP neurons in this region. And vice versa, of the AGRP cells, not many of them were activated by the food cue. When we looked at the ghrelin receptor, this is interesting because this information comparing FOS and ghrelin tells us whether the same cells are activated by the food cue as by ghrelin, so by an external and an intrinsic orexigenic stimulus. And again, a little bit to our surprise, the numbers were not higher, but we still saw something like 30 to 40% of the cells were co-expressing cue-induced FOS and the ghrelin receptor. This is just confirming essentially some old data where we know that most of the AGRP cells in this region express the ghrelin receptor, and of the ghrelin receptor cells expressing AGRP, yeah, it's slightly over half. So if it's not AGRP, what else could it be? And so this is just the statistics again for AGRP. I've just shown you about 20% are AGRP cells. We looked at another obvious candidate group in this area, the pro-opiomelanocortin cells. These are actually, conversely, an anorexigenic cell group in this area. And we found about 40% of those cells are ghrelin responsive. And finally, we did a triple RNA scope for FOS, tyrosine hydroxylase that signals the dopamine cells in this area, and the ghrelin receptor, and we found that only about 12% of the cells activated by the food cue were this tyrosine hydroxylase population, and most of these were ghrelin responsive. So around something like a third then of the cue-activated cells appeared to be ghrelin responsive. So conclusions then, I think I would like to have shown you more data, but we certainly have plenty data backing this statement that ghrelin is both a hunger and an appetite hormone, and therefore intrinsic signals such as ghrelin and external sensory food cues drive food intake and food-motivated behaviours. The arcuate is targeted by converging orexigenic signals, so about a third of the cue-activated neurons are ghrelin responsive and vice versa. The cue-activated arcuate cells are heterogeneous. They include both orexigenic cells, anorexigenic cells, and some dopaminergic cells, but we still don't know almost what a half of the cells are that are activated by the cue. And finally, external sensory food cues trigger ghrelin release, perhaps gaining its status as the aperitif hormone or perhaps an appetizer hormone. Appetite and hunger may not be so different. After all, if you think of these rats in that cage going crazy trying to get the hidden food, what are they doing there? Is this behavior because they're hungry? Or is it because they have an appetite for peanut butter? Very hard to distinguish between them. And of course, the fact that we have some convergence of signaling suggests that appetite and hunger may not be so different. And we've provided a biological basis that food cues make you hungry, releasing ghrelin and engaging so-called hunger pathways. So coming back to my original slide, I think the fact that the arcuate nucleus is targeted by food cues means that we could also place an arrow from perhaps more reward areas back to at least the arcuate nucleus in the hypothalamus. So on that note, I just thank my lab, my collaborators. For this study, especially Fiona Perez San Pedro is the driving force of this project, and Iris Stoltenberg, a PhD student in the lab. Thank you.
Thanks a lot. That was really a good appetizer for the day, and I myself have a lot of questions already, but we will leave them for the end of the session. Now use the app, send your questions, and it would be really good if you could also put in brackets who you want, an answer from, so that I can. There will be easier for me to then ask each speaker. Then we go on without further ado, and the next speaker is Aniko Korosi, Associate Professor at the University of Amsterdam, and she will talk about the entrée, the long-term effects of micro and micronutrients on cognition and stress resistance. Thank you.
Right. Oh. Thank you so much for the nice introduction, and I'm very happy to be here. It's really nice to finally give again presentations in person. It's great. This one. So we will switch a little bit gears, and I will talk to you about the importance of the early life period, and of course, the importance of nutrients during this early life period. I think I don't need to convince this audience that the first 1,000 days are key determinants of our mental health later in life. And with the first 1,000 days, we often encompass, of course, from conception up to about the first 2 years of life. And indeed, when adversities occur during this sensitive developmental period, then this has negative consequences for the vulnerability to develop psychopathologies later in life, such as cognitive deficits, depression, but also many other psychiatric disorders. So when we look into the epidemiological literature about what leads to these negative consequences, what is striking is that, of course, the adversities can take many, many different forms, often more psychological, for example, like neglect, but also physical, like maltreatment, but also in the form of malnutrition, for example. And when we look at the phenotypes that are being found later in life in these individuals that are exposed to these adversities, they are remarkably similar. In addition, it's nice to realize, or it's important to remark, that these psychopathologies are really often comorbid with metabolic disorders, and that indeed these early life adversities also lead to these metabolic disorders themselves. So there is a lot of interaction and intersection that it's definitely worthwhile to explore further. So in the lab, we really try to understand what are then the key elements on this early life environment, which can be so diverse, determining these phenotypes, which are then quite similar with each other, and which are the main neurobiological substrates, and how do these different elements interact. So some years ago, we started to think about the different interactions and the different components, and we decided to focus on the interaction between stress and nutrition, and how these early life components, the early life stress with early life nutrition together might interact with each other and lead to alterations in brain plasticity, and thereby also in the phenotypes, in the behavior, in the cognitive functioning, but also, for example, in anxiety-like behaviors. And how will these two interact? Also, of course, with in mind the idea of finding some targets that are actually possible to implement also in actual human populations. So what we use in the lab is a mouse model of early life adversity, which is of limited nesting and bedding material. It consists of exposure for the first postnatal week. So it's only 1 week of exposure to an impoverished environment. So the mother is consistently present, but she simply has like a more erratic way of taking care of her pups because she doesn't have all the facilities that the other mom has. She has less bedding material and no nesting material. And we know that this leads to altered plasticity. This has been shown by us and by many, many other labs that use actually this same model. And it leads also to cognitive deficits and changes in anxiety-like behaviors. So starting from this, we pose the question, are nutrients then involved? Can nutrients be involved? And if so, then how? And can we use this as a possible tool or target for interventions? So why nutrients? And of course, what attracts me a lot about nutrients is that, of course, they are building blocks of all our membranes and all our cells, but they're also very important signaling molecules. And this is the aspect that I actually would like to tackle with you today. So it's important to realize that, of course, I'm going to talk about a really hardcore preclinical model, but there has been a really increasing interest in nutritional psychiatry and how we can use nutrition to modulate actually mental health. And what we realize more and more is that while this is really attractive and it's really promising as well, and most of the studies are so far associative, and we still need a lot of information on the causality between them and the underlying mechanism. So to be able to really take all the opportunities from these kind of strategies, we need to learn a lot more about it. ECMP has also put a lot of attention to this. Indeed, we have also the ECMP Nutrition Network, which is a really specialized network focusing specifically on these questions. So the questions that I will be specifically tackling is, okay, does early life stress alter nutrient levels in the brain? Can nutritional intervention prevent these early life stress-induced effects? And then which are the mechanisms for that? So the students in the lab that actually did all of the work that I will be presenting are Dr. Kitty Yam, Kitty Reims, and Marilyn de Abing. And later on I will also mention a collaborator. And the concept is just that. We have early life stress. We have some known phenotypes that we already have characterized earlier. Can we understand what is the role of nutrients in this, and can we intervene with nutritional interventions? Then, of course, the next question, which nutrients are we going to tackle? And the ones that we decided to focus on are essential nutrients. So nutrients that we need to take in from the diet, and we focus on essential fatty acids and essential micronutrients. So let me start with the fatty acids. We know that fatty acids, I think you're all familiar with the importance of taking omega-3, eating salmon and nuts and olive oil instead of other forms of oil, for example. But actually, what is really important to realize is that the ratio of this omega-6 and omega-3 fatty acid is what is really, really important. It will be key determinant of brain development, of properties of your membrane, for how, for example, membrane fluidity is, of neurogenesis, and it has really important inflammatory modulatory roles as well. So earlier, about a few years ago, we have shown that an early diet intervention from postnatal day 2 to postnatal day, I don't manage to move the mouse for some reason, but doesn't matter. Oh, yes, now it moves with me. Now, in any case, we have this early life stress exposure, and we have the diet from postnatal day 2 to postnatal day 42. And we have shown, first of all, that early life stress actually reduces or changes fatty acid composition of the brain, which is interesting by itself. That this early dietary intervention manages to rescue these fatty acid alterations. What you see here, this is moving by itself now. Okay. And we have shown that this early life dietary intervention is able to prevent the cognitive deficits that we have induced by the early life stress. This is here shown with the object location task, but we have done that also with other tasks. And also some of the changes in brain plasticity that we have observed earlier. So the changes in adult neurogenesis, it was in the form of neuronal cell survival, but also changes in the neuroimmune cells, in microglia, in the CD6 microglia CD68, which is a phagocytic marker of microglia. So what you see in these three graphs is indeed, I don't know why this is moving by itself now. I will go back 1 second. So what you see here is that early life stress indeed leads to deficits in behavior and plasticity, and that this can be fully rescued by an early diet. So the diet is long gone by the time that we look at these phenotypes. So we went further and we wondered, how does early life stress and this early diet affect other aspects? So we wanted to understand, how does the diet do this? How do we reach these beneficial effects? And we decided to explore the peripheral cytokines, but also the gene and lipid profiles, both under basal state, but also with a secondary immune challenge with LPS, because we think that really often these secondary challenges are really important to unmask possible latent effects. So this is quite a complicated design, and you will hear me about several of these comparisons, because this is, of course, is an 8-group design at the end. And indeed, within these animals, we looked at the peripheral cytokines, at hippocampal gene expression, and in the hypothalamus, we looked at the broad lipid profile of the hypothalamus. So what we learned is that, first of all, yes, LPS, of course, alters cytokines, there is no surprise there, but that early life stress modulates how this response to LPS is. Early life stress by itself increases some of these cytokines, for example, IL-6 and CXCL1 and CCL2, but also there is an interaction. So this early life stress aggravates some of these responses in cytokines in response to LPS. This was quite a harsh LPS, I must say. In hindsight, we should have probably could have been better using a little bit a milder LPS injection. This is also why we have such a plateau in LPS response. But nonetheless, we see some interesting effects. So what happens to gene expression analysis? Does the diet impact on it under basal condition? Does it affect on it in response to LPS? And is this dependent on early life stress in any possible way? So I will just show you a little bit of this data, of course. So first of all, we see that an early diet, which is long gone, impacts on the gene expression profile of the hippocampus. And what we see, for example, we see, of course, many different geoterms, but we see, for example, indeed changes in long-chain fatty acid transport and long-chain fatty acid catabolic processes, kind of confirming that this diet indeed somehow might be incorporated in the hippocampus. So this is just a comparison between controls and under the two different diets. And what you see here with these Venn diagrams is that you can compare, of course, the comparisons. So you have the controls and early life stress under the high diet, so with a certain ratio of fatty acids, and the controls and the early life stress under the low diet. And what is striking to see is that indeed, in both cases, there is, of course, altered gene expressions, but that these are almost entirely different. So depending on the diet, we have an entirely different set of genes that are being altered by early life stress. One remarkable one that will come back also later is, for example, that under the low diet, which is the one that actually rescued our early life stress-induced phenotypes, is the extracellular matrix, which is one of the top hits. What happens in response to LPS? So LPS induces also gene expression changes that are different in controls and early life stress mice and depend on the diet, of course. So what we see again is that we have a completely different set of genes depending on the diet and depending on early life stress and on the LPS. And again, even in response to LPS, what we see with the arrow is that under this low diet, in response to LPS, the LS and ELS animals exhibit changes in relationship to extracellular matrix, which might be then an interesting target to approach. So what happens with the hypothalamic profile, lipid profile? So this is also something that we tackled in this study with a really similar comparisons. And I will just give you a glimpse of that. What we see is that this diet that was given early in life, many months later, actually alters the lipid profile that you find back in the brain, which is maybe also to be expected, but still it was really interesting to see it categorized as such. And what we see is that it decreases hypothalamic lipid species implicated in inflammation and stress. We also see that this low diet ratio differently modulates the lipids in controls and early life stress animals. So again, we see that there are not that many lipids changed, but the ones that are changed are different depending on the early diet. And last but not least, what is really striking and interesting is that LPS, just in response to LPS, we actually alter our lipid profile in the hypothalamus, and this is highly dependent on early life stress and on the dietary ratio. What you can see here with this more complicated Venn diagram. So I think in summary for now, we see from the hippocampal gene expression that this is affected by early life stress, of course, by dietary ratio, under basal level, and also in an immune challenge condition. And that may be one of the hits for how these beneficial effects of the diet is, which was actually the question that we had to begin with, might be related to the effect that it has on extracellular matrix and transmembrane receptors. So this is something that we are currently following up. Similarly, the lipid profile was affected by all of these different conditions. So what we are currently doing is actually studying further the gene expression analysis, also with the microRNA data set, together in collaboration with Dr. Cattaneo from Milan. And we are also looking at not only at the lipid profile, but also the SPM profile, so the specialized pro-resolving mediators, to understand even further how the signaling aspects of these fatty acids are. So just a glimpse of what we do with the micronutrients, because that was also part of the goal. But the micronutrients that we focus on are the ones that are belonging to the one-carbon metabolism. So they are really important for methylations, for producing methyl groups that are important for DNA methylation and for histomethylations. And these are, for example, folic acid, vitamin B6, B12, etc. So again, in a study of a few years ago, we have shown that an early supplementation with these micronutrients is able to to rescue some of these phenotypes of the early life stress. First of all, early life stress changed the methionine composition of the brain, it reduced it. And what we see is that with the diet, we managed to rescue these methionine levels, we managed to rescue the cognitive deficits. And what was really interesting and striking to us is that the HPA axis seemed to be modulated by this. So while we have normally an increased corticosterone in response to this early life stress, this was completely absent in the presence of this micronutrient. So this led to our next question, which we did then in collaboration with Veronica Begni from Milan and Kitty Reimst. We supplemented the same diet as before, but instead of waiting to adulthood, we looked in adolescence, and instead of only looking at basal level, we also looked in response to acute stress, again with a similar principle as with the LPS earlier. So it's a quite complicated study again with many groups, and we submitted the animals to a long battery of tests to really tease apart the anxiety phenotype of this model. We exposed them to elevated plasmase, the dark light box, the splash test, and sucrose preference test at the end. And what we see basically is that indeed the early life stress leads to changes which are often apparent only in response to acute stress. In this anxiety phenotype, so elevated plasmas and dark light box by early life stress are mostly affected in response to acute stress, and this is rescued by the diet. The splash test was a little bit more obvious already at basal level, but also in response to acute stress. And again, this effect was modulated by the diet. We did not see any effect on the sucrose preference, but this was also quite far from the acute stress exposure itself. So we think that that's actually the reason for that. What we did next, we wanted to see, okay, our main goal with these micronutrients was actually to modulate, hopefully, possibly, the epigenetic machinery. So in collaboration with Moshe Sif, we went and we did a genome-wide methylation analysis in the hypothalamus to understand what are the effects there. And what we found was actually very interesting. First of all, most of the sites, 86% of the sites are hypomethylated by acute stress in control animals alone. This is what you see in these first two bars. Early life stress alters the methylation levels upon acute stress. This is what you see in this comparison. So this is really, really interesting. And many of these sites actually are on sex chromosomes, on the X and Y chromosomes, also implicated in neurodevelopment. And last but not least, what we were also, of course, really happy with is that the diet reversed most of these effects that were induced by early life stress. So the animals that have received the micronutrient early in life, during the adolescence, exhibit a very different methylation profile. We, of course, looked at the relationship of these methylated sites with the behavior, and there were some that were also correlating, 74 the differential of these different sites also correlated with behavior. So I think I will skip the summary because I'm already a little bit over time. One last slide that I would like to show you is that, of course, we are also intrigued by where does this different nutrient composition comes from upon the stress. And one of the hypotheses that we are pursuing is the possibility that the stress exposure of these dams affects the maternal milk composition. And we are doing that actually in a human study. And we have the first results on the fact that stress indeed correlates with the altered fatty acid composition of human milk. And in addition, bringing back also to the presentation of Suzanne, we also have found that early life stress affects actually food choice later in life. So it's possible that another additive value is also the type of choices that an early life stress individual makes later in life for what they eat and what they don't eat. So just to wrap it up, I would like to one more highlight that animal models of early life stress are translationally very valuable, that next to stress hormones and sensory stimuli, which have received a lot of attention in this context, nutrients play a key role, that nutritional interventions are very promising and could be a new potential avenue, but we need to learn more. Nutrients could also serve as a contributing factor to the search of the biomarkers for vulnerable individuals. So we need to understand further about the mechanism and how all of this interacts in order to be able to optimize the current nutritional strategies. I thank you for your attention, and of course I thank all the collaborators and everybody that did the work. Thank you.
Thank you very much. And we go on now with, I guess, going to a different animal model. I think this will be very interesting to see how these mechanisms that we have seen so far can be applied in an animal model like Drosophila. And this will be, the speaker will be Carlos Ribeiro. He's actually group leader here in Lisbon at Champalimud. And his talk is the PLAT, Nutritional Decisions in Drosophila, including the impact of the gut microbes. Thank you.
Okay, thank you very much for the kind introduction. It's also great to be here finally in an in-person meeting and not staring at a screen. So today I'm going to, I think, give a bit of a more exotic talk for this setting and tell you about our work in Drosophila and trying to understand how brain-body interactions shape nutritional decisions in the animal. So I would like to start by thanking my team. Oops, okay, no, this is not, okay, the mouse is really not working. So I would like to thank my team, a fantastic group of people who have, whose honor I have to be the spokesperson. Also thank our collaborators. I think working on nutrition and the brain requires a lot of interdisciplinary work, and it's really thanks to our collaborators that we can really span work from behavior to metabolomics to genomics and many other things. And then finally also our funders who keep us and our flies fed. And here I would really like to emphasize and thank the Champalimaud Foundation. This is our center. It's the Champalimaud Center for the Unknown. It's a state-of-the-art cancer and brain science institute, which if you walk here along the river towards the ocean for 5 kilometers, you can go there. And I think on Monday there's going to be a very nice ALBA reception there, so I encourage you to go there. So if you think about what most neuroscientists are interested in, or most of the work actually has been done in neuroscience about food, is the question of how do animals decide to eat or not to eat. And I think while this is a very interesting and important and extremely productive field, I think if you think about it, what the animal really needs to solve is the problem of which nutrients to eat in which amount, right? Because every different nutrient has a very different impact on health and behavior. Energy has a very different impact than vitamins, and proteins and fats also have different impacts. And so how to really reconstitute a complete and optimal diet from these different types of opportunities is something which I'm really interested in. And we're focusing our work mostly on amino acids and proteins, right? So if you think about it, amino acids are obviously essential, right? We cannot make proteins without amino acids. And in contrast to other nutrients, we cannot make 10 out of the 20 amino acids, so they're essential. And they are actually more or less the same essential amino acids in all animals, including Drosophila. And so that means that we have to actually eat them to be sure that we have enough of them. But interestingly, actually, just overeating amino acids so that you're sure that you always have enough amino acids is also not an optimal strategy. Because I think work over the last 20 years has shown in almost all animals ever studied, and including epidemiological studies in humans suggest the same, is that overeating amino acids and proteins shortens lifespans, as has a very negative impact also on health span, right? So we need to eat enough amino acids, but we cannot overeat it because that will shorten our lifespan. And so we want to understand how the brain deals with this trade-off and solves it. Now, thinking a bit more translationally, I think there's also a lot of interesting evidence that amino acids have a very strong impact in neuropathologies in the brain. And here I want to just highlight one study from my colleague Gaia Novarino, who's now at IST in Vienna, which has shown that in certain families where children have a very severe cause of autism spectrum disorders, there are mutations in the genome which all seem to impair branched-chain amino acid uptake and metabolism in the brain. So there seems to be a link between amino acid access to the brain and neurons and neurocognitive aspects. So if we think about what are the factors which determine food choice, we and other people have mapped out multiple aspects of that. For example, right, reproductive state in women and also insects and rodents impacts how much and what type of food, especially amino acids and salts, the women but also animals will eat. Another important aspect is the metabolic need of specific organs. So organs speak with the brain and through hormones and peptides instruct what they need and will change the food intake and food choice of animals. But one thing I want to emphasize today, and I'm going to discuss a bit and focus today, is the impact of the microbiome. So I think there is an increasing awareness that our gut microbes have a strong impact on brain function and also neuronal pathologies, right? But we are really at the beginning of understanding what are the mechanisms by which actually gut microbes impact brain function. I think one very attractive hypothesis which I would like to highlight in this audience, because that's a bit the theme of today, is the idea that microbes in our gut obviously interact with the diet and nutritional components in the diet and modify them and manipulate them. And if you think that, or if you believe that nutrients have a strong impact on the brain, then obviously this interaction of gut microbes with nutrients, including the production of nutrients by the gut microbes, will then act on the brain. And so this is an interesting and I think very attractive hypothesis to think mechanistically about how gut microbes could affect brain function. But there are many, many open questions which I think we need to tackle as a field, right? First of all, how do nutrients shape the microbiome? Why do we need communities? Because I think one thing which has emerged also is that it's not single bacteria which affect physiology, but it's communities. Why do we need bacterial communities? And then just very, very importantly, obviously, what are the mechanisms by which gut bacteria change behavior? And I think there Drosophila can be a very attractive model. First of all, Drosophila has a very conserved genome, so molecularly Drosophila is very similar to rodents, vertebrates, and also humans. And here especially metabolic pathways and regulatory systems like insulin, leptin, etc. are conserved. But also the organization of the physiological systems like organs are also conserved, right? So the digestive, the body organization, the nervous system, including also metabolically active organs, are very similarly organized and function between humans and insects. And I'm sorry that I cannot move the pointer here to highlight some things. And I think the power of Drosophila is also highlighted by the multiple Nobel Prizes which have been given over the last 100 years to work which has been done in flies. And here I would also like to highlight the last Nobel Prizes which are related to neuroscience, the discovery of chemosensory systems, innate immunity, and also circadian rhythms, which all have been shown to be conserved from flies to humans. But what do we know about the microbiome in flies? Well, flies have a very simple microbiome. If you catch Drosophila in your kitchen, you're going to find mostly 5 types of bacteria. Lactobacilli and Acetobacter are going to be mostly prominent, similarly than we also find in invertebrates. And they have been shown to have a lot of different impacts throughout the whole lifespan of flies, from development, where they're very, very important to promote growth, to adults, where they change lifespan, tissue homeostasis, and feeding preference. And interestingly, the same bacteria can have the same impact on vertebrates. So if you take the Lactobacillus plantarum, which promotes growth in Drosophila, and you put it into a mouse, which has been kept in malnutrition conditions, you're going to be able to rescue the malnutrition effects on growth in mouse by putting in the same bacterium which has been isolated in flies, suggesting that there's a functional conservation, right? So, but we want to understand food choice. So the way we measure food choice in flies is using the fly pad. This is the same principle as you use on your iPhone and iPad if you're bored and now are reading your emails instead of listening to me. And what it does is it measures the touch of the fly with food, and from that we can measure every single food intake event from the fly. So what is it we want to really understand, right? So if you look at 3 different types of flies, flies which have been kept on a full diet, flies which have been kept on an amino acid deficient diet, and flies which have been kept on a carbohydrate deficient diet, and you give them the option to eat from amino acids or from carbohydrates. So this is like a buffet choice option. Then what you're going to see is that flies which have been kept on an amino acid deficient diet, they will specifically increase the intake from amino acids, but not from carbohydrates, whereas carbohydrate deprived flies will only increase the intake from carbohydrates and not amino acids and proteins. So they're able to identify a lack of specific nutrients and then compensate by eating from that nutrient. And humans do that too. So if you take people and put them on a low protein diet for 2 weeks and then give them the option to eat whatever they want, you will see that they will overeat proteins, most likely to compensate for the lack of proteins. So the brain is able to solve this problem, right? To eat right, it's able to detect that it's lacking specific amino acids or nutrients and then compensate by choosing them from the diet. And I think this is very, very at the basis of understanding nutrition and how the brain controls nutrition. But let's get back to the microbiome, okay? So this is a classic experiment we would do with our flies in the lab. So you have flies kept on a full holytic medium. So this is a fully chemically defined medium in which we can manipulate every single nutrient. And we're comparing flies kept on a holytic medium, which has all amino acids, to a fly which has been deprived of only one essential amino acid, which is isoleucine. And what you see is that there's a drastic increase in protein intake. Yeast is the main protein source for flies when they're isoleucine deprived. Now this is seen mainly in flies which don't have a microbiome, so in germ-free animals. If we put back 2 specific bacteria which are found in wild flies, Acetobacter pomorum and Lactobacillus plantarum. Now these flies, even if deprived from isoleucine, will not be eating protein. And you need both bacteria, because only putting in Acetobacter pomorum or only putting in Lactobacillus plantarum does not suppress protein appetite. So these two bacteria have the ability to suppress protein appetite in the flies. So the question now, how do gut bacteria change food choice, and why do you need a community of two bacteria to change behavior? And here what we have been using is isotope-resolved metabolomics. So isotope-resolved metabolomics allows us to trace in vivo in the fly or ex vivo in culture the route by which specific metabolites which are marked with non-radioactive heavy isotopes are modified by the bacteria or by the animal. And so what we can show, and it's too bad I cannot point here because it's a bit of a complex slide. So what you're seeing here is the amount of isoleucine being produced in culture in conditions where you have either only Acetobacter pomorum or Lactobacillus plantarum, and we are only looking at the amino acids produced from labeled glucose. And what you can see is that you need both bacteria to be present to produce significant amount of isoleucine in culture, and we have also new data showing that the same thing is in vivo. And we can trace that back to lactate, and so what happens is that lactate is being produced by Lactobacillus, and so if you put labeled lactate alone together with Acetobacter pomorum, you can see that now these bacteria produce high amounts of isoleucine on their own using the lactate we put from the medium. So does this matter in vivo, right? And so another thing we can do in flies is we can do bacterial genetics in the animal. So what we can do is we can mutate the ability of Lactobacillus to produce lactate and check if this is important for changing behavior. So again, here, the control experiments. If you take flies, remove isoleucine, they increase protein intake. Acetobacter pomorum alone doesn't do anything. Acetobacter pomorum with Lactobacillus now suppresses protein appetite. And if we mutate the ability of Lactobacillus to make lactate, we cannot get a rescue. That's the second last bar there. And we can rescue that by putting back lactate in this mutant bacterial background. So the question is Acetobacter and lactate sufficient to change behavior? Again, we can take now a condition in which we have Acetobacter alone, which doesn't suppress protein appetite. And if we add lactate, lactate together with Acetobacter pomorum is sufficient to completely suppress protein appetite in these animals, and that's not because lactate has specific effects, because if you put lactate alone in there, there's no effect. So lactate is a key bacterial metabolite being produced by Lactobacillus, which is required to change behavior, and Acetobacter is the key bacterium altering behavior. Now, I won't show the data, but it turns out that this is really important because the reason the Lactobacillus need to produce lactate is so that Acetobacter pomorum can make amino acids, and this allows Lactobacillus to now survive in the gut of the host because Lactobacillus cannot make isoleucine, okay? So that means that there is a circular nutritional economy there, and this metabolic cross-feeding allows the gut bacteria communities to remain stable even if important nutrients are missing. But the amino acids are not what changes behavior. And if you want to know why we get to the conclusion, you need to read the paper. I don't have time today. Furthermore, this also shows us that Acetobacter is the key bacterium which changes behavior, and that lactate is a precursor to produce psychoactive substances. And I think this highlights the power of having complete control over the diet and the use of isotope-resolved metabolomics to now look what are the metabolites produced by Acetobacter from lactate, and that's something we're doing now. And to understand what happens in the brain, we can do whole brain calcium imaging in the fly. This is what you're seeing here. So this is a fly brain now with GCamp. And we can see the fly brain neurons, all the neurons in the fly brain, how they are activated when the fly is eating. So we can watch the brain of the fly while it's eating. And we can do that in flies which have or don't have a microbiome, or flies in which we reconstitute the metabolome of the fly by putting in specific metabolites. So I propose here that because nutrients have such a strong impact on behavior, that by looking at neuronal circuit function, molecular mechanisms involved in nutrient sensing in the context of nutrients and the microbiome, using quantitative behavioral assays, putting that in the context of phenotypes like aging and health, and taking into account metabolism and organ physiology, there at the center is really where we can start to understand mechanistically how nutrients act on the brain and how body-brain interactions shape brain function. And obviously that's best done in flies. And this is not only just for food intake, because if you compare flies which are fully fed with protein-deprived flies and look at how they search for food, you see that they do use very, very, very different strategies when exploring the environment and looking for food. Again, showing that the nutrient state of the animal, and here we're talking specifically about the protein state, affects cognitive-like behaviors which are obviously related to foraging and therefore are involve complex cognitive computations. These are the fantastic people who have done the work. If you're interested in working with us, would be the view from your working place. And I invite you to watch our talks. We have every Monday a seminar series, a virtual one on brain-body interactions, in which we discuss these type of phenomena, which we think is actually very relevant for all kinds of pathophysiological understandings of brain function. Thank you very much.
Thank you, Carlos. That was really fascinating. And now we are only missing the dessert. And the dessert is served by Giulia Chiacchierini. She's a postdoc at the Institute of Technology in Italy. And the talk is The Dessert, Neural and Behavioral Analysis of a Nutrient-Specific Appetite for Protein.
So we finally arrived at the final course of this tasty symposium. I would especially like to thank the organizing committee for having me today, giving me the opportunity to replace my PhD project supervisor, Jamie McCutcheon, who couldn't make it today. And indeed, today I'm going to show you some of the data I have collected during my PhD project. So in case you were wondering, this beautiful animal behind me is a Peruvian spider monkey. So let's imagine for a second the monkey in its natural environment, the subtropical forest, surrounded by a huge diversity of food resources. Sometimes these resources are scarcer, sometimes more abundant, depending on many factors, including, for example, the time of the year. But importantly, what the monkey does, regardless of the food choices available, the monkey will always select food in order to reach a daily specific protein intake target. So the monkey is willing to under or overeat carbohydrate and fat if this means reaching the protein target. In other words, if the food available is rich in protein but low in the other two macronutrients, the monkey will eat not much of that food, decreasing the energy intake. On the other side, if the food available is very low in protein but rich in the other macronutrients, the monkey will overeat that food in an attempt to reach the protein target. So this animal is an extraordinary example of the fine regulation of dietary protein, which is also called protein leveraging, and which has been demonstrated across different species, including humans. And in humans, in particular, protein leveraging has been proposed as one of the causes of diet-induced obesity. So in the original paper, the authors presented in the USA over a period of almost 40 years. They showed that the concentration of carbohydrate and fat in the diet has increased by a lot, but the proportion of protein instead has remained pretty constant. And the authors remember that in the same country, in the same time range, the incidence of obesity has increased by 3 times. And the authors also showed the change in percentage protein in the diet against, it doesn't show here, but on the y-axis there is the percentage incidence of obesity in the adult population. And they found a negative correlation. So for example, in countries such as Denmark and the Netherlands that saw an increased percentage of protein in the diet, the obesity rate is at the lowest level. So starting a little bit from here, in the lab we were wondering how does the brain ensure appropriate protein intake? And we started looking into that by placing some of our rats on a protein-restricted diet containing 5% protein. We were looking at the average food intake of the animals, and we saw that the protein-restricted rats in green increased their daily food intake compared to non-restricted control rats. So this suggested us that a sort of protein regulation was taking place. But it's interesting also to see that the body weight of the two diet groups remained similar. Then the lab developed its own model of conditioned protein appetite. So 2 weeks after the diet manipulation, rats underwent a conditioning session. So in this block, rats were given either a protein-rich solution, casein, or a carbohydrate-rich solution, maltodextrin, on alternate days. Importantly, each solution was paired with a specific distinctive flavor. For example, cherry for casein and grape for maltodextrin. So during this conditioning block, rats had the opportunity to learn about the post-ingestive consequences of consuming casein or maltodextrin on their physiological state with a specific flavor. Then a preference test followed in which both solutions were available at the same time. Rats could choose which one to drink, and the number of licks made for each solution were recorded. And we saw that during the preference test, protein-restricted rats in green made a significantly greater amount of licks for protein compared to carbohydrate. And this preference was evident also by calculating the casein preference score, so the number of licks for casein divided by the total number of licks. Then we started wondering how important this conditioning phase was in order for protein appetite to develop. Because in literature there is still debate whether protein appetite is something that needs to be learned by the environment, by the association of post-ingestive consequences with food cues, or instead protein appetite is something innate, somehow inborn in the organism, a little bit like what happens with sodium appetite. And to have an idea about that, we decided to remove the conditioning block from our behavioral paradigm. So 2 weeks after the diet manipulation, rats went straight to the preference test. And we saw that already during the first preference test, protein-restricted rats, always in green, made a greater number of licks for casein compared to maltodextrin. Then we decided to look at the time course of licking behavior within the 60-minute session to get an idea about whether rats could be learning about post-ingestive consequences within the session. And we see that non-restricted rats in gray made a similar number of licks for casein and maltodextrin throughout the session, while protein-restricted rats already since the very first minute of exposure to the two solutions started preferring protein over carbohydrate. So this suggested us that maybe previous experience with nutrients, it's not really essential for protein appetite to develop. Then we started wondering whether a state of protein restriction would lead to changes in cognitive processes, in particular in the motivation towards food. And we investigated that using a progressive ratio schedule of responding, which is a classical paradigm used in neuroscience, in which the number of lever presses needed to get a reward increase exponentially after each reward delivery, until a point at which a rat doesn't press the lever anymore because it requires too much effort. And this point is called break point, and it's our measure of motivation. And we did this task in 2 different batches of animals with 2 different reward types. So first with pellets that had a balanced composition of the 3 macronutrients. And we saw that with nutritionally complete pellets, the motivation to obtain food was similar between non-restricted and protein-restricted rats. But when protein-only pellets were used, we observed an increased motivation in the protein-restricted group. Then we started wondering what happens when the protein-restricted rats are replated. So they are put back on their control regular chow diet, and after a week they are tested again on the progressive ratio task for several sessions. And we saw that already during the first progressive ratio task, protein-replated rats may add a similar motivation compared to rats that never experienced protein restriction. So this was indicating us that the increased motivation towards protein is rapidly dismissed when it's not needed anymore. Then we finally wanted to see and get an idea of what are the neural substrates that contribute to driving protein appetite. And we did so taking advantage of fiber photometry, which is a technique through which you can measure real-time neural activity while animals are freely behaving. So we injected a genetically encoded calcium indicator in the ventral tegmental area of our rats, and we placed an optic fiber in the same brain region. So through fiber photometry, we were able to measure calcium fluctuations as a proxy of neural activity. And in our paradigm, we used a system of retractable zippers. So you will see in the video that when the light cue goes on, this signals that the zipper is now extended and available. And the rat starts leaking, and after 5 seconds, the zipper retracts. And here we can see the response of VTA neurons to the cue and to leaking behavior. So behaviorally, we found that during first choice trials, meaning when one zipper or another was available, we found that protein-restricted and non-restricted rats made a similar number of leaks for casein and maltodextrin. Then we looked at free choice trials when both zippers extended at the same time and rats could choose which one to drink. And we saw that here, again, protein-restricted rats made a greater number of leaks for casein compared to maltodextrin. So first choice trials were important because they gave us the demonstration that rats did not develop an aversion towards carbohydrate when they were on a protein-restricted diet. If we then looked at the neural activity, we see that in non-restricted rats, there is a response to the leaking for casein and maltodextrin. But this neural response is similar for the 2 solutions, as indicated by both the Z-score and the area under the curve. On the other side, when we looked at the neural activity in protein-restricted rats in response to leaking, we see that protein-restricted rats have an increased neural activity in VTA when they leak for casein compared to maltodextrin. So to sum up, today I showed you that rats are able to develop a strong preference for protein compared to carbohydrate when they are protein-restricted. This preference seems to develop quickly at first experience with protein food and involves changes in the motivation for protein. Moreover, we have seen that the activity in BTA evoked by each solution is closely matched to behavioral preference. I would like to thank my former supervisor, Jamie, that now established his own lab at the Arctic University of Tromsø in Norway, everyone that helped me with the project, and thank you all for kind attention.
Thanks a lot. I think this was really very interesting, and now I have some questions from